Can you hear and see? Well, obviously you can see because you're saying. All right. So before I start, then I know I say this every time, but I'll say it once more time. The fact this is the last week, so the final exam is scheduled for the 21st, 2:30 in the afternoon, I believe. I forget which room it's in, but you have got a choice between a sort of sit down face to face, we'll call it, and a virtual. If you want to take the take home exam, then you're going to tell me by Friday at midnight. On the day after, I'm going to send out example questions to everyone else. And just to remind you about the virtual exam, it's going to be released on the 18th and it's going to be submitted at the midnight on the 21st. And then the following day, we're going to have a normal exam, about 10 minutes. And it's just the material, regardless if face-to-face or virtual, the material not covered in the midterm. So it's not going to include the midterm material, but it's going to contain everything other than that. Is that all right? I'll take that as a yes, I guess. That's good. And we'll probably get very close towards the end of this lecture of the material. This is the last unit. On Thursday, if you want, we can do a revision class. So does anybody want a revision class? Oh, you do? That's good. That's the way. Me and you will be here then, at least. Oh yeah, somebody else does as well. So it's all right. So yeah, so I don't. I'll take it easy today, and we might get to the end, might not, but we will definitely get to the end on Thursday. We've got a revision class then, so you can bring your questions. I can bring some questions if you like, and I'm sure, but together we'll have questions. All right, that's good. All right, so we are coming to the last unit now, and that's to do with strain selection and improvement. So, one of the universal things we've had is throughout the units is that the microbes are at the heart of any fermentation or any process. So the actual process of fermentation, when we get a new isolate, it's going to be pretty low productivity. It's not going to have all the desired traits. And so what we have to do is domesticate it. Domesticate it to do what though? So like I say, we'll go out and get a new isolate from somewhere, be it a tree, if we're going to make food science beer. But what do we need to improve on it? So it comes undomesticated, if you want to call it that. But what's the aim of strain selection improvement? And you can answer online as well. All right, fair enough. Well, we want it to be productive. We want to increase the productivity. When we talked about penicillin, if you remember, mutations to make it better. When we talked about penicillin, we know that when it was first isolated, it took a whole year to produce enough penicillin just to treat a half a dose for one person. Obviously, by increasing its ability to produce, we could get to a point where it was producing 1,000 times more than it originally did. And we do that by improving it. We need to improve stress sometimes, get rid of the undesired traits. And so that's the way to think about as we go along, how we improve strains. But the first thing we have to do is obviously isolate them. So what we're going to be doing is talking about how we go about getting new isolates. And then we'll be talking about how we can improve them. And the reason why we want to domesticate microbes is because if we rely on this spontaneous fermentation, like we talked about for various, like sauerkraut and all that, Christian Hansen, he came up with the pure strain brewing for a reason. It was consistent. It was desirable characteristics in terms of the flavor profiles. And obviously, when we talk about amino acid production, we talk about yield, growth yields, product yields, and we have to improve its stress because we put cells through quite an ordeal when we're talking about fermentations, especially when we talk about biofuels, isn't it? Because they're always recycled to save on biomass, and therefore we get stressed. And obviously, one of the big things is novel products. A novel food product, a novel drug, a novel surfactant, a novel enzyme. That's what gets the head, doesn't it? So where do we get microbes from? So they all start somewhere. And in the golden age, which we call the 1950s, even I wasn't born then, people would go anywhere in the world, to the Arctic, to the Amazon, to the hot springs, to the volcanoes, to the everywhere, just to isolate microbes. And it was this sort of expanding knowledge on new microbes that we discovered, which had special characteristics. And so when we look at for microbes, we have to think what we want to use them for. You can go out there on a wild goose chase and say, well, I'm just going to find a microbe. I don't care if it's antibiotic production, I don't care if it's biofuel production. But usually we have an aim to go there and find a new drug or find a new enzyme. So what we'll do is sometimes we know what the genera does. So if we think about antibiotics, for example, what sort of microbe would you choose, go out looking for? So if we're looking for a new antibiotic, we want to be a Nobel Prize winner, what would we look for, do you feel? All right, fair enough. Well, Streptomyces is a good one to choose this because we know Streptomyces produce over 60% of the antibiotics. So if we're going out hunting for a new antibiotic, it may be good to look for that first. But there again, we might say, let's just go for any antibiotic. And we'll talk about metagenomics in a bit. So that's one part of it. We just go randomly to different environments, and we probably choose our holiday destination, saying, oh yes, I feel like going to the Amazon or something. But there again, we can look for specific niches because we know that microbes have evolved for specific niches. So if we're looking for a cold loving enzyme, say we're making a cold detergent, well, we don't want to go to the thermal springs, we want to go to the Arctic. Try and get an enzyme that works at cold temperatures. Sometimes we want hot temperatures. If we think about biofuel production, we look at gelatinizing the starch and degrading at the same time. So you need a thermophilic enzyme, so you go somewhere hot. So I think you get the idea. But rather than send our people out to different parts of the world, what we could also do is go to culture collections. So if you ever discover a microbe or a new species, you have to characterize it, then deposit it in the culture collection. So in the US, it's ATCC, which is the American type, American type culture collection, or something like that. And every different parts of the world have their own culture collection. We'll talk about it a bit later. So what we have to do then is really isolate the microbe we're interested in. And typically, we either know the genera, as in we say, well, we want Streptomyces, or it could be just a brand new microbe, something nobody's discovered before, a bit like when they discovered the new antibiotic producer. But the thing is that you're literally panning for gold. These microbes are hidden in a big population, and some of that population is non-culturable. If we think about the Tioxobaxin, that's really was unculturable when they first isolated it. So it's a bit of a job, isn't it? Because you're trying to pan for gold on this big background of microflora to find this one microbe that will produce your special antibiotic, or overproduce this amino acid, or whatever you're trying to do. So what we have to do, isn't it, is trying to be selective. Is there a selective pressure for it? And when it comes to, for example, looking for, if we're looking for something that can degrade cellulose, because we want to get into cellulose now rather than corn bioethanol. If we're looking for cellulose, that's easy, because in a way, what we can do is provide cellulose as the only carbon energy source, and then we put it into a chemostat, and the only microbes that will be able to stay in that chemostat before being washed out would be the ones that can metabolize cellulose. And so that's one aspect, it's a competitive advantage. You could also develop media, which is just cellulose as the carbon and energy source. Again, the only microbe that will grow is the one that can degrade cellulose. So this is, and even if you're talking about a toxic substrate, you could think, well, if we introduce this toxic substrate into the media, like a precursor for an antibiotic, and the microbes grow, that means it's detoxifying it. So I think you get the idea. If it's selective pressure, that's easy, because you can just use that as a benefit to pan the gold, to get to that microbe you want. But there again, all the interesting things are secondary metabolites, the antibiotics and things like that. And that's a bit harder for the simple reason is that they're produced under non-growing conditions. So you can't have selective pressure for that. So you have to go for this laborious art of getting microbes, isolating them, testing them for whatever antibiotic, a protein inhibitor, drug, whatever you want to do. And it's just screening assays. And this what takes the thousands of isolates. So it literally goes day after day to do. And it's very expensive. So as I say, secondary metabolites, they're the most interesting because they kind of evolved to be more complex for some odd reason. So if we're doing antibiotics, you have to do challenge testing. If we're doing protease inhibitors, which are usually drugs for HIV and whatnot, they're just enzyme assays. And if it's a binding assay for a receptor on a drug, you have to do that. So if you like, you're going to go for all these thousands of different samples, just doing the assay to find one that actually works, which you can imagine very, very laborious, expensive, and even then you've got to try and find the microbe after that. So, but things have changed, and we'll talk about how they've changed a little bit. So that's kind of the gold rush, if you want to call it. You're basically just going out there to isolate. So you don't know what the microbe is, but you're going to try and isolate a producer. And that's basically what they did for the Trioxabaxin. If you remember the iChip, we literally put these microbes in a chip, dug it under the ground, and just waited to see what would grow and produce antibiotic. That's how they found it. But as I mentioned, the other way of doing it is to be targeted. So we know Streptomyces, for example, is that most of them produce antibiotic, which is good. So what we can do is go searching for them because we know the chances of getting a good one is quite high. So for that, we need to develop media. So we all know selective media. We've used it in the lab where you basically selecting for the microbe of interest. So when we think about foodborne pathogens, we look at, for example, with Salmonella. Well, we know it's got bile resistance, because some bile in there. We know it's very tolerant to low water activity, so we put high salts in there. And we know it produces it produces ammonia from the degradation of amino acids, so we can put I think it's ferric sulfate in there, and we get a distinct colony. So basically what we're saying is that we can be selective, keep down the background microflora, and differential, be able to tell the microbe of interest from those in the background. And also what we can do is add an elective agent, i.e. something that promotes the growth of the target bacteria. So when we talk about Streptomyces, for example, we know it's very tolerant to histamine, whereas other microbes aren't. So we can add histamine to the media. We know it's got not a unique feature, but one of the unique features, it can degrade dextran. Dextran being a polymer, as you know. So we can use that as a carbon source so other microbes can't. And we can also put antibiotics in because Streptomyces can't inhibit itself, so it's naturally antibiotic resistant. So what we're doing there is selecting for them in terms of keeping the background microflora down. We can add growth stimulating agents, which are going to enhance the growth of the Streptomyces. And we can differentiate it because Streptomyces produces very distinct colored colonies when it's under iron deficiency. So that's just an example, but regardless of what you're doing, you've got to kind of develop the media depending on the carbon source, depending on its physiology, so you can bring it out from a very complex sample such as soil. All right. But there again, why do we have to bother with all this? Why can't we just go to a culture collection like the ACTC and buy a $300 culture and no need to isolate, no need to do anything? Well, it's true. Every country's got its own culture collection. Some are 3 or 4 of them. And like I mentioned to you, if you isolate a new microbe, you typically characterize it, you name it, and then they'll say you can't name it after yourself, you've got to name it after something else. And then you deposit it in a culture collection. So even if you're developing a probiotic and you find a new one, again, you've got to give it over, not give it over, but I suppose it's the done thing. So you can go to the culture collection and look at all these Streptomyces isolates if we were talking about that. And you can just buy them for about $300 a time. And you hope that you're going to get something special. Well, it's a bit like going to Value Village and hoping to find a Rembrandt. It doesn't happen very often because obviously if it was special, the people who first isolated it might be able to detect it. But you never know, because obviously they won't, if they're not looking for antibiotic production, I might miss it. But that's a kind of one way of doing it. But then you get into this issue, and I'm pretty sure there's been a few cases here, is that who owns the IP? So if you get a Streptomyces and it produces brand new antibiotic worth billions of dollars, if it is worth billions, the culture collection will say, well, that belongs to us. And if you read if you do buy an isolate, they've got a whole page of saying you're not allowed to keep it after your study, you're not allowed to swap it. Obviously people do. But it comes to a point saying, well, who owns the IP? Is it the original person who isolated it? Is it the culture collection itself, or is it the person who discovered it? And there's a few court cases there, but culture collections have protected the bag by saying, no, it belongs to us. So keep quiet if you ever hit. All right. So obviously, like I say, in the golden age of microbiology, where literally for your PhD you would go out to all these exotic locations, some nice, some not so nice, to isolate new microbes. That was the golden age. But it soon became apparent that we're missing a lot. Essentially, most of the microbes we encounter in nature are non-culturable. They don't play the game. And this is called microbial dark matter. So, and I suppose I'd say dark matter to make it look physics or something like that. But essentially what it's saying is that even in soil, only about 5% of what you see can be cultured. Because if you think culturing in the lab is very artificial, it's giving it a nice sort of media which you think will make it grow. But a lot of microbes need this interaction with other microbes. They're not naturally present on their own. They've learned to build associations up, which is one reason. But we're missing a lot. We're basically missing 95% of the microbial population. And you could debate saying, well, it's all right even isolating something that can't be cultured, but how are you going to make it ferment? But we'll worry about that in a little bit. So what happened in the 1990s, towards the end of the 1990s, people came up with this approach called metagenomics. So back in the 1990s, genomics was a bit, what's the word, primitive, if you want to call it that. We didn't have very sophisticated techniques. We ran gels, we ran southern blots, western blots, northern blots, and things like that. But the concept was there. The concept is that a lot of the microbes we encounter in nature can't grow, so we can't characterize them. But what we could do is use something called environmental DNA, as in the instructions to prepare a new product or encode for a new drug, antibiotic, whatever you like, is written in the DNA. So what we can do is take that DNA from a whole population, we can search it, so we can read the copy number, read the actual DNA itself, and then we can use a big computer to decipher the sequence and also identify sequences of interest, identify sequences which are present in every gene of that sort of ilk, i.e., for example, I'm explaining it badly at this time. If we're looking for antibiotic genes, obviously non-ribosomal peptide synthesis is the sort of key genes, enzymes that's going to be present. Now, all NPRs have what we call a conserved region, i.e., they're present in every single NPR, and the only variation, obviously, is the other genetic coding. So if we go looking for these conserved regions in a sample of soil, for example, which we can't culture, then the chances are that we can find this gene, bring it out, put it to something else, and be good. So long story short, metagenomics is looking for DNA within a mixed population. We call it eDNA, environmental DNA, where we take the whole DNA, we chop it up, we make what we call a clone library, which I'll talk about, then express it. And then express it so we can detect it and then go forward from there. And also what we can do with this technique is look at those networks we're talking about. The whole population, how microbes interact, how they get partnerships, and how they cooperate or don't cooperate. Now, in the 1990s, as I mentioned, we didn't have sophisticated technologies. It took 4 years to sequence a whole genome, and that's what would be your PhD. You could do the same within a week now because of the sequencing technology. So what we had to have is enabling technologies such as DNA sequencing, data processing power, the data storage, and also the knowledge. People back then didn't know anything about metagenomics. So it's even today, it's kind of, well, what you would say, I would say it's still in the early phases, but by the time you graduate, it's probably going to be fairly mainstream. Anyway, let's see what we're doing. So as I say, metagenomics is this technique where we look for the DNA. We're not looking for the product, we're looking for the DNA signature of an enzyme, of an antibiotic producing enzyme, things like that. So we've got two choices. We can do a sequence analysis, i.e. we're basically panning for gold in these sequences. We're getting all these sequences and we're hoping our sequencer and a computer program can identify those conserved regions, saying this sample has something special in it. Or we can do what we call functional analysis, where we try to take the whole genome, not whole genome, the whole gene, and insert it into a vector, which then will produce an enzyme, will produce an antibiotic, things like this. So this is it, functional analysis versus sequence analysis. And as you probably guessed, sequence analysis is much easier. All right then, so as I mentioned to you, this is coming online, right? Well, I would say about 5 years ago, it became more routine, let's put it that way. So let's see what we're doing with metagenomics. So what we know is that we can't culture the bacteria from there, or the microbe, for various reasons. So we'll get a sample. Yeah, we'll send someone to the Amazon, send someone outside. Well, we get a soil sample, for example. Now, what we'll do is extract all the DNA from that sample, and then we use restriction enzymes. So restriction enzymes are these scissors for DNA. So they'll cut at specific points, and there's lots of different restriction enzymes, which we'll talk about later on. So you cut in different places. Now, as I mentioned, the reason we do that is because we know we can't grow the cells up. So, but we get this DNA, we cut it up, and we put it into a vector, ligate it. So obvious one is plasmids. So we can ligate or cut up pieces of DNA into this plasmid, and we'll insert it into E. coli, because E. coli has been the workhorse, the better or for worse, as we'll talk about later on. And then we transform it. So we're going to, and I think you can see where we're going. We put the plasmid into the E. coli, and then we've got a choice. We can make this clone library, and we can do sequence analysis, which basically means that we grow the E. coli up, obviously copies and copies, and then we digest the DNA, and we'll put it onto a southern blot. So a southern blot detects DNA by hybridization. So we'll have a primer or oligomer, which would hybridize after it's separated from the gel. And if it's there, it'll light up, if you want to call it that. If it's not there, it won't. But there again, you see the product of it in that if it's, for example, a conserved sequence for a non-ribosomal peptide synthesis and it lights up, you know the original sample has that target in it, that thing you want to look for. Now, I don't know if you've ever run a southern blot. I think this is the old technology in the 90s, I suppose. Northern blot's been RNA, I think it is. Very laborious. So, but now it's sequencing. You know, you literally got machines that can do it in no time at all. So that was an enabling technology. So it's the same sort of principle, though. You'll grow up the E. coli, you'll express the gene, and then what the sequence does then is this program, the data processing will look for that conserved region of interest to say there's something here, or more or less most of the time there's nothing here. So that's sequence analysis, but functional analysis is where you take longer cuts, if that's the right term, longer cuts of the DNA to try and get the whole gene. Not only get the whole gene, but have it functioning. And we'll talk about how difficult that is in a little bit. But if you get the whole gene and it's functioning, say, for example, you're looking for an enzyme to produce a degrade cellulose or an enzyme to cold wash your clothes or whatever, what you try to do is express the whole enzyme which is functioning, and then it will basically be transported out of the cell. Like I said, that is striking gold because it really happens. And the reason why I say E. coli is not a very good expression vector is because it doesn't obviously do all the things that gram positives can do. It doesn't do all the things eukaryotics can do. So some people say E. coli wasn't the best, but anyway, we won't go into that. But is that clear to everyone, or is it a bit confusing? Right, fair enough. So activity-based is where you're basically trying to get the whole gene, which is very, like, to actually be able to cut so specifically on an unknown target is very challenging. So it has that disadvantage of you'll really get any hits. But if you do get a hit, you've got the gene intact. You know, you basically can cut it out, characterize it, insert it into E. coli or whatever, and express it to get that protein you want. Now, sequence-based analysis is basically just, as I say, the conserved regions of the gene. You know, if you're looking for a thermophilic amylase or whatever, you broadly know what the sequence is, and you can either hybridize it if you're doing southern blotting, or you can ask your program that's going to go for all this sequence data. If you see these conserved sequences, raise the alarm. So typically what would happen is it would do the sequence base first and then move to the activity base, obviously, just for the pre-screening. And as I mentioned to you, this is quite new technology. It might sound, oh, wow, this has been established for years. It hasn't really. And the reason for that is because if you think about it, 5 years ago, that's when our first DNA sequences really came. Even food science food science never did go. Pathology did, or microbiology did. About 5 years ago, I think it was when the first DNA sequences actually came available to normal labs, I'll call it. So it's quite a new area. And like I say, it takes all the fun out of going to the Amazon and things like that. But after saying that, people have used this approach and will use this approach to find novel enzymes, to find novel drugs, find novel products. And these are just some of them. So BASF, that's a German company, they discovered an amylase, which was thermophilic. And obviously, we use amylase bioethanol production. Phytase, they're reducing the phosphate in manure of pigs because obviously that leads to pollution of water and things like that. There's polymerases, polyketide for antibiotic production, and lipase and things like that. So again, they did this metagenomic approach rather than culturing. So just to go through that then, what we have in metagenomics is functional sequence approach. Functional approach is where we get the fragment of DNA, which is a whole gene, if you want, a functional gene. We insert it into a vector and hopefully we'll produce the product of interest. But as I say, it's like literally it's like hitting gold. Not only do you have to have the gene there, but you've got to cut it in the right place. You don't know what the sequence is between the flank, and you've got to get all the promoters and things like that. So with sequence-based, though, it's much easier because you're basically just looking for a particular conserved sequences, and therefore you're more likely to take it. But once you have that sequence, all you know is that sample contains something special, but you still have to do you have to find where the gene is and try and work it out from there. So as I mentioned to you, it's becoming routine now, and you're looking for new antibiotics, so you can look for the polyketide sequences. For a long time, when we're looking at non-ribosomal peptide synthesis, the thought was saying, well, we don't need to go looking for new ones. We can just use genetic engineering to modify the ones we have. And it's quite interesting is that when they try to modify the sequences of pre-existing non-peptide synthesis, they usually have lower yields or don't work very well. So, nature has the way, as to say, it's going towards perfection. And we use it to look at enzymes, and we also use it to study these sort of behaviors. We talk about the microbiome, don't we? In terms of this interaction of the microbiome to produce beneficial effects to health, stimulate moods and things like that. Well, the only way we can understand that is to express the 16S RNA of these cells and then look to see what populations are there and how those populations interact. Because as I keep going on about, the cells don't live alone. It's a bit like us. So we all live together. We all interact most of the time, sometimes beneficially. So thinking about that, one of the big applications of metagenomics, other than looking for drugs and enzymes, is looking for the microbiome, but more specifically, what we call probiotics. Basically these microbes that gives us benefits. In a lot of ways, lactic acid bacteria have taken over the mantle of probiotics. The reality is they're not very good probiotics, but people like lactic acid bacteria for simple reasons. Say, oh yeah, yogurt, it must be good for us and things like that. But anyway, but there are additional ones, but we won't confuse the matter. Now, probiotics is a big market, worth billions. People take them every day, tablet form, yogurt form. And before a few years ago, basically, you could bring anything out and call it a probiotic. As I mentioned to you, it was a case, forget Activia, I think it was. They actually made up a name for a probiotic. And even though there was no probiotic in there, I forget what the name was, but they basically made the name up and said, this will make you feel better if you take 14, give it for 14 days. So this Wild West sort of probiotic market had to be brought to heel because people were making claims that weren't justified, saying, oh, it causes low blood pressure, it will make you healthy and all this. So the FAO, which is the global organization, agricultural organization, WHO, we all know about WHO and the pandemic, isn't it? World Health Organization, came up with a definition of a probiotic, and that was basically any microbe, when administered, will give benefits. So if you think about it, you say, well, what's benefit? How are we going to prove what benefit there is? Because there's so many confounding factors. So what you have to do, though, if you want to get a new probiotic, which is not on the probiotic traditional list like bifidobacterial, lactobacillus, you've got to prove a benefit. And that's quite hard, to say, well, our probiotic does this. So there's various targets that can choose, though, to show benefit. So one of them is a bit like I've always said, and we've come across this quite often, is the competitive inhibition or competing for nutrients. So acetic acid bacteria would be world champions at this, if only they were anaerobic, because obviously the GI tract is anaerobic. So what this means is that they can outcompete or oututilize, I suppose the word is, the growth substrate. So before your competitor can get it, you've taken it. And acetic acid bacteria, as we talked about in vinegar production, are world champions at this because they can take glucose, convert it to gluconic acid, and deprive the competitors of that nutrient. So that's one way of doing it. So the second way of doing it is, I can't really see the numbers, is producing bacteriocins, or bioactive peptides, as they call them, or making fatty acids, some of which are antimicrobial. So this is a direct antagonism. You're basically beating down the competition by producing these antimicrobial peptides and things like that. What you can also do is produce what we call prebiotics. So prebiotics are substrates that enhance the growth of good bacteria, whatever good bacteria are. So for example, you might produce butyric acid. That's a popular one. You might produce vitamins, biotin, something like that, which promote the growth of beneficial bacteria. Now, as I mentioned to you, the definition of beneficial bacteria is up to conjecture, but I think you get the idea. You can also have direct antagonism. So basically, this is various mechanisms where they can inhibit the microbe or make the pathogen, if you want, because I guess we don't know what good bacteria are, but we still know what bad bacteria are in terms of pathogens, those cause infections. So some produce proteins, which are called aggregating factors, which would bind to the pathogen. And basically, if it binds to the pathogen, it tells the immune system, saying there's a pathogen here, come and take it out. So that's direct antagonism. And obviously, the other ways of doing it is producing acids, lactic acid, acetic acid, and producing hydrogen peroxide. So these are all direct antagonism towards the pathogen itself. I'm trying to look for number 5 now. Oh, here it is. So number 5 is competitive inhibition. So this is a bit like when it takes on nutrients. It's basically outcompeting the pathogen. It's outgrowing it for various reasons. But what it can also do is line the epithelial cells to stop the pathogen binding, because obviously pathogens want to get into our bloodstream and things like that. But what happens is if you're probiotic, and we talk about lactic acid bacteria, as I said, because of the nature of the sort of concept, it can colonize the lining of epithelial cells, therefore not allowing the pathogen to do the same thing. This one's more, not controversial, but people debate about it. I can't even, I think it's number 8. Anyway, go number 8 and go to number 7. This is stimulation of the immune system. So we all know about vaccines and COVID vaccines and other vaccines. What we do is we kind of give a small dose of a foreign protein, and that stimulates our immune system to produce beta cells, antibiotic producing cells, and T cells, the ones that produce cytokinins to activate the immune system, which produce memory cells. So basically what we're saying is that if the immune system encounters that protein again, it's ready to fight. It's not going from ground zero. That's how vaccines work. And so what the thought is that certain probiotics produce proteins that resemble that of a pathogen. Obviously, it doesn't have all the virulence factors, but it stimulates the immune system to produce these antibodies and memory B cells in order to tackle those proteins if it's encountered again. So stimulation of the immune system, again, some people believe it, others don't. But there again, what you can do is down-regulate the immune system. So some conditions, when we think about toxic shock syndrome, it's basically our immune system being enhanced, basically being triggered for no apparent reason. And and once our immune system's triggered, it's got neutrophils, and the neutrophils go and destroy cells. It produces cytokinins that get metabolism, fevers going. So what can happen with certain probiotics is it can down-regulate the immune system, to stop it, self-destructing. So all of these are basically targets for probiotic bacteria. So when you want to say benefit, you've got to prove kind of one of these actually is working. And we'll talk about how we do that in a bit. So as I mentioned to you, you've got a choice. You can go out and get a new probiotic and say, well, look at this new probiotic. Or you can go to the Health Canada website where they've got particular species or genera and species, which are automatically classed as probiotics. So say, for example, you went to find a Bifidobacterium bifidum, and you could identify it as that because you use the 16S RNA and things like this. Then you say, yeah, that's a probiotic because we know the strains belonging to that species have probiotic characteristics historically. A bit like a Lactobacillus acidophilus. If you get an acidophilus strain, so yeah, it's must be the same as all the other acidophilus, so you can have that. And so you'll get the probiotics, and what you have to do is prove that at the point of consumption, it's got 10 to the 9 CFU per serving. So as we talked about in yogurt production, these aren't very good acid producers. These aren't going to be involved in the main fermentation, but we add them afterwards, encapsulated to preserve them in order that they're 10 to the 9 when people eat them. And as I say, if you think 10 to the 9 is a lot, when you think how many cells are in the GI tract, it's literally a drop in the ocean, but it's cells, so why not do it, isn't it? But, if you want to, that's all right, but you say, well, if I just use one of these probiotics, I'm just going to be the same as Activia. I'm not going to have that sort of advantage. So there's a lot of interest in finding new probiotics, and Emma in microbiology has done this. She's, I forget what they call that food pill. Anyway, she's got a business making probiotic tablets. I forget what it's called, but anyway, so but they're not lactobacillus. They're something else. I think it's guarded secret. But anyway, so how do we do it? Well, we do exactly the same as we did when we looked for new isolates. Regardless of it's producing antibiotics, glutamate, anything like that. You go to the source of where you're going to encounter the benefit you think they're going to be. Well, it goes without saying, if we get one from a GI tract, well, that's already adapted to the body. So there's a good chance that it's going to be a good probiotic, a chance. If we turn off our nose and say, well, we don't really want to start taking fecal samples and whatnot, we can go to fermented foods because people know fermented foods. It's a bit like, say, yeah, sauerkraut and all this. I can relate to that. We can go to milk because we know lactic acid bacteria, for example, exist in milk, and milk has beneficial bacteria. That's kind of proven. Everyone knows that if you give Clostridium to animals and humans, it establishes the good GI tract, ones that aren't prone to pathogens, things like that. Anyway, so you get the picture. We go to the isolation source of where we think our probiotic will be. And then we get specific media, as I said to you. People say, oh, lactic acid bacteria, MRS, that's good. The reality is, though, is we might have to get specialized media. So if we're going to do specialized media, what do you think we need to consider? Nubota, that's an interesting one. So what do we do? Say if we're developing a media, how do we go about it? All right, fair enough. Well, first of all, we have to think about compositional analysis. Do you know when we develop media for fermentations, where we do this composition analysis, say it's got this much carbon, this much nitrogen, these ions. So that's the first thing. The second thing is we look at its carbon utilization profile. Is there anything in there that's special, which will be able to select against another, i.e. promote it? We might try and recreate the GI tract, bile salts, for example. A lot of lactic acid bacteria, very sensitive, and gram-positives in general, are sensitive to, so carbon source, the nitrogen source, pH resistance, bile resistance. So we develop our media based on that. Then once we've got our isolates and we've screened them, we look at the 16S RNA. And we do this for two reasons. One, obviously, we need to know what it is to look at it. But we can do the whole genome sequencing to look for virulence factors. So if you remember when we talk about Wessler, which is in Leuconostoc family, it's a very good starter culture. It's high acid producer, but the problem with it seems to acquire antibiotic resistance. It has hemolysin as a virulence factor. So whole genome sequencing, you can look for these antibiotic resistance, and you can also look for virulence factors. And if they're there, well, maybe this isn't the one for you. You have to go to the next one or try and sort out what they are. So once it's passed that test, as in no virulence, then you look for the benefits, or, well, I call it safety, I suppose it's true. Is it antibiotic resistant? You don't want antibiotic resistance because you're going to cause infections, not fight infections. And it is a bit controversial when you think about it, because if, for example, you look at Clostridium difficile, multiple antibiotic resistance, and we get Clostridium difficile when we're on antibiotics. And you would think, well, why don't you just put a probiotic, which is also resistant to antibiotics, to fight it off? But, it's not a done thing. So you can't have antibiotic resistance. And the virulence factors is you do not only look for it on the genome, but you do animal trials. So if you administer this probiotic, you discovered it doesn't cause more problems than solve. And then you start looking for benefits to it. Now, you look for, well, will it resist the stress of the acid and the bile? And to the benefit, would it adhere to the epithelial cells to cause that blocking of pathogens? So if it kind of ticks everything, saying, well, yes, it's not going to kill you, it's not going to become resistant, it can survive in the stressful environment of the GI tract, It then goes down to the next one, which is basically also showing benefits. So does it prevent infection from Clostridium difficile, from Salmonella, from Clostridium perfringens? And does it modulate the immune system? Does it upregulate it? And thinking about it, if you like, when trying to prove a benefit, it's much easier to say, oh, it's immunostimulating, because you can measure the B cells, the somatic cells, and T cells say, look, we administer it, these go up, therefore it must be good. But when you come to say, well, it inhibits salmonella colonization or Clostridium perfringens intoxication, that's harder because there's so many confounding factors. You know, is it the host susceptibility? Is it the dose? Is it the environment? But anyway, you've got to show some benefit to go to the last stage, which are the clinical trials. And as we talked in antibiotic production, clinical trials have 4 phases, you know, going from a small group right through to the mass population. The vaccines for COVID kind of passed about 3 of those phases, but anyway, that's another matter. But you've got to show benefit in terms of, yes, it's safe, yes, it reduces blood pressure, yes, it makes people feel better. So as you can imagine, bringing a new probiotic to market is very difficult, very expensive. And you think about the rewards, well, you know, maybe it is a big market, but you know, people like the yogurt and people like the lactic acid bacteria, I suppose. So as I mentioned to you, what you've got to, once you bring a probiotic, and I suppose the riches are waiting for people to do that, you've got to prove it's safe, you've got to prove that it gives benefit. And also what you have to do, as I mentioned, if it's a brand new species and strain, or genera, should I say, or both, you've got to deposit in a culture collection. So automatically, you're kind of giving away your silver, but you've got to do it. And the culture collection will say, well, yeah, it belongs to us now, and things like that. But anyway, but you can see to actually put it on a label is very challenging. And you'll notice these days, that unlike the old times, is that they're very cautious about what claims to make. They don't make it direct claims. They just say, well, take activity, you'll feel different. Doesn't mean say you'll feel good, it just feels different. Anyway, I'm digressing. All right then, so we have our isolate. And as I mentioned to you, once you've got your isolate, you've got to domesticate it or train it, whatever term you want to use. And we have strain improvement. So typically, and we covered this in the first unit, I think it is, industrial strains, and you know, I suppose you could include food strains on that, but food fermentations are a bit different. They're more artistic things. If we think about bioethanol production, or if you think about amino acid production, I guess it's easier. So what we need from industrial strains is they need to be stable. You know, you can't have ones that just mutate frequently and change the characteristics every fermentation, because you want every fermentation to be bang on. You want efficient production of the target. You know, the economics are very slim. If it's not producing maximally, you're losing money on it. And if you're losing money, you're not going to be in business long, is it? And the only way to get efficient production is know the pathways. We talked about pathways, how we modulate them and modify them as we go along. And therefore, we need to understand what pathways there are in order to do that. We need to have some simple nutrition. And we think about biofuel production with Xymonas. Xymonas produces much more ethanol than Saccharomyces, but we can't use it because it just hasn't got the substrate utilization profile. But yeast are good, you know, because they'll take anything almost, isn't it? So we need to be able to use a sort of cheap media to grow them on. And this is why cellulose producers and cellulose utilizers are such big interest, because they're basically living on wood. They've got to be genetically manipulated. They need to be very well understood on how they function, how they work. And as I mentioned to you, everybody put a lot of effort into E. coli because that's been a workhorse. But as we talk about later on, it may be we took the wrong decision there, but something had to be the number one. So you need to know the genetics well and how it all works. Non-toxic and pathogenic. So when we talk about biomass production, Candida is actually much better for biomass production, if we're making alternative protein sources and Saccharomyces. Saccharomyces has too much RNA, produces gout, and not so good, sore foot. But the trouble with Candida is that it's pathogenic, and therefore you have to take its pathogenic traits out to use it. We can't have 20,000 litres of pathogenic yeast going around. And a bit like when we talked about enzyme, cellulases production, it needs to be exported. We don't want to go into the cell and having to extract it. We want everything to be exported, be it amino acid, an enzyme, or things like that. So these are the traits we look for and try to improve upon when we get a new isolate. So we have to improve it because we want to make it much more stable. We want to make it much more productive. And by doing so, make it much more economical to use and go within that cost feasibility. So the targets for strain improvement are essentially what the ideal characteristics of an industrial strain is. We need to have it to high growth rates. We want to get the fermentation turned over as fast as we can. We need it stable. We can't have it mutating, so we can look for improving that. Obviously, remove the virulence factors because we don't want pathogenic traits. If we can kind of take them out, it makes it easier. We can put genes in. We talked about cellulases being inserted into Clostridia and also inserted into Saccharomyces in order to be able to use cellulose. The reality is it's not working very well, but you see the point, trying to modify the strain in order to utilize these substrates. And minimize byproducts, not only byproducts that would interfere with purification, but to increase efficiency. We talked about lysine production. We literally cut a branch off the pathway to overproduce lysine because we don't want the carbon going through these other sources. So that maximizes production. And as we talked about in lysine production, what we have to do is get rid of the negative feedback or feedback inhibition or repression. So what we do? Well, we do select a different carbon source, but we basically mutate the aspartate kinase to not be inhibited. So it's like a runaway train. So we do that by mutation, deregulation, and we could export the target, make sure that protein is exported through the type 4 secretion systems and things like that. And also, if you think about bioethanol production, what we want to do is increase the ethanol tolerance because we want to produce as much ethanol per batch, but we don't want to kill the yeast. So how do we do that? And as we talked about at length, we modulate the carbon electron flow to overproduce what we're interested in. If we have to cut branches off metabolism, that's all well and good, but we need to get as much productivity as possible. So when we're looking at strain improvement, obviously what we need to do is going to happen through mutation of some kind. It's basically the wild type is going to be the parent strains, and we're going to have a overproducer. And that's going to be by a mutation, which deregulates an enzyme, enhances activity or turnover rate or things like that. And there's different ways of approaching this. So we could do natural selection. So Mandel and his peas, I suppose the word is, if you remember that. That was natural selection. So basically saying, oh, well, breed this one with this pea and then we do that, so we'll get a better pea, whatever it is. So that's natural mutations. All right, if you've got a long time to wait and all that, but typically, obviously what we do these days is genetic manipulation, GMO, if you want to call it that, and mutagenesis. When we think about mutagenesis, we've come to the new age now of CRISPR, and we'll talk about CRISPR in a little bit. So with natural selection, it's a bit like being in the class. So you've got really well-performing people. The main body of people do all right, and one or two struggle. But the thing was, if we take the top of the class and then keep subculturing it, in terms of producing antibiotics, for example, they're going to get better and better. They're going to push each other, I don't know. But anyway, the point is, and I think it's a bad analogy, is that within a population, you have hyperproducers, and if you select for them, it goes without saying that you'll increase the productivity. And that's exactly what they used to do. Now, if we're looking at a selective substrate, not antibiotic production, say we're looking for, say, a cellulose, enhanced cellulase producer, well, let's put it in a continuous culture and turn the wick up on the dilution rate, because it's only those that will survive and grow if they can utilize the cellulose quickly. So it's a very laborious process, as you can imagine, and you're really just depending on nature. So what you have to do is give nature a helping hand sometimes. And we do that by mutating agents. Now, the most popular, I suppose, if there is a popular one, is UV. So we know UV is bad for us because if we go into the sun, we get damage to our DNA and things like that. And that's what UV does. It damages the DNA. And now it can damage it on a positive scale. Like most of the time, it's going to be negative. It's going to be lethal mutations that render the cell unable to produce anything, never mind live. But there again, sometimes you get positive. A positive mutation, a deregulation of an enzyme, may be a switch in the amino acid to make it less substrate specific, a bigger turnover, less sensitive to feedback. And that's called the hyperproducing strain. So we can do this by exposing it to UV light, or we can use agents such as acridine orange. So acridine orange is a carcinogen, and all you have to do is make a gradient plate. So, and you can use any toxic substrate, because a lot of toxic substrates affect DNA, as you probably know. So we use these gradient plates where we have our sort of acridine orange, if you want to call it that, on one side. And we pour the plate so it's at an angle, so we get a nice wedge. And then we add unsupplemented media on top. So we have this gradient. Obviously, here it's very high concentration mutagenic agent. Here it's very low. And what you'll find is a lot of the population will grow in the low sort of concentration of a mutagen because it's not doing anything. But then you'll get some which are very resistant. And they're the ones that probably have altered the DNA somehow in order to, and it might be beneficial. But as you can imagine, it's very non-specific. It really is. Like you're not changing one piece of DNA, you could be changing lots of different genes at that time. But this is how they did it back in the day. You know, they would literally get, for example, if you're doing antibiotic production, you'll get your spore suspension of your streptomyces, and you'll mutate it, UV light, exposure to acridine orange. Those that survive, you place onto the media, and then you'll take some colonies. You'll always keep a backup because you don't want to lose what you've done. So you'll keep a backup of select few, and then you'll see how they produce the antibiotic. Is it good yield? And then once they get the good yield, and as you can imagine, it takes such a long time to do these assays, if you think about it. But then you take the top 50 producers and do the process all over again. You know, life as a microbiologist back then wasn't fun because it would be just literally repeat, repeat, repeat all day long, all week long, all month long. But those old methods, you know, essentially did fairly well. You know, basically, as we talked about penicillin, when it was first isolated, it took a whole year to produce half a dose. By 1972, where I think even your parents weren't born than I was, it was up to 1,500, and you think today is probably much higher than that. But it just goes to show you these sort of basic techniques can produce results eventually, but you could imagine it, well it's going to be a bit of effort to get to there. So as I say, with to enhance nature or give nature a nudge, we can use these mutagenic agents to speed up the natural selection process. But again, you know, it's very non-specific. You know, it's basically just mutating all these things, hoping we'll get a good one, where most of them are going to be fairly lethal mutations. So it went without saying, say, well, can't we do it a bit more selectively? Oh, we've got sound effects even. So what we can do to get more selective is try and target specific regions of the DNA. Now, as we talked about with UV lights and chemicals, it's very non-selective. You're going to mutate more genes than ever. So what we need to do is look at the book of DNA sequence, which is the genome, and find out which genes to mutate and do it specifically, because it's going to be more efficient and we know what we're mutating. We're not globally mutating the genome. We're just going to take out or change that gene of interest. And you did your first year chemistry or biochem or whatever what is. The DNA is the code book. It is our sort of directory of how we make proteins, and those proteins become enzymes, transporters, all these kind of things. So you know that for good. So site-directed mutagenesis was developed in the 1970s, and these are very old techniques, and it's well, some people say it's outdated already. It's one of those old techniques that we always relied on, but when something better comes along, like CRISPR, it stays a done. But anyway, I'll go through it because you never know if you'll ever come across it. So with site-directed mutagenesis, there's different ways of doing it, but essentially what we're doing is being selective in the gene, even the amino acid we're going to change in this protein. And people took their whole PhD to figure out what one amino acid sequence does. I don't know how you do a PhD on that, but it's everyone to their own, isn't it? So what we can do with site-directed mutagenesis, as I say, selectively alter proteins by changing a genetic code which encodes for them. We can also revert it back because we want to mutate the gene, but we also want to revert it just to prove, saying, yeah, when we got this amino acid there, it does this. When it hasn't there, it does that. And one of the techniques then is cassette site-directed mutagenesis. And with this technique, what we're doing is that we're getting our double-stranded DNA from a cell, E. coli, yeast, whatever you want to do it. Then we melt the DNA to make it single-stranded. So when it's single-stranded, we then get our sort of oligomer, which has got a sort of hybridization. It hybridizes the target because we know the sequence we're looking for, because we've done DNA sequencing using our Sanger sequencing, whatever it is. And so what it'll do is hybridize, but it won't hybridize perfectly because one of the base pairs is different. It's not matching up, and you know about AT and GC, I'm pretty sure you do. So what the cell does, you say, well, all right, then we'll just improvise and we'll try and repair it and we'll copy it. So it copies, the DNA polymerase copies this one, and it makes two versions, obviously. It makes a wild type version, which is what it should be, but also it'll have a mutated version with this different encoding base, which basically encodes for a different amino acid. And so you have your wild type, your natural protein, which might have valine there, and it might have a mutated type, which has alanine. So it's altered. And what they try to do is obviously look for the active site, look for the regulatory sites. If we change this amino acid, it does this. If it changes that amino acid, it does that. And like I say, people used to spend whole PhDs just looking at this sort of thing, making it sound interesting. But it's a very laborious process, as you can imagine. Is that all right for everyone? Yeah, okay. Yeah, this is just another sort of version of it. So basically, it's, as the name suggests, we melt the DNA, we add a primer, which isn't exact pairing, but then it does replicate, and it replicates, so producing a new genetic code. And a new genetic code means that we've got a new amino acid, which means we've got different functionality. There you go. Very laborious technique, but it basically was, back in the day, was what people did. So it's all right having this tool, but what they realized very early on is saying, well, which enzyme should we mutate then? Which gene? And as we saw in biobutanol production, you're naturally thinking, oh, well, the alcohol dehydrogenase must be the rate limiting step. So if we increase the alcohol dehydrogenase mutatis, and we'll get more bioethanol. No, it turned out the ferredoxin, which produces the NADPH, is the rate limiting. If we think about glutamate production, it'll be a natural thing to say, well, if we increase the glutamate synthetase, we're going to get a better reaction, more productivity. Well, no, you've got to upregulate pyruvate carboxylase. So anyway, to the point, looking at which gene to mutate or which enzyme to mutate isn't straightforward. And this is where microbial physiology comes into play. And when we talk about BioBricks, we'll talk about why that's important. But if we take lysine production as an example, because that's a very good example of how we had two challenges. Well, we have three, but the two main challenges, we have this branch point, which takes carbon to threonine, methionine, and we also have another branch which takes it to lysine, which we want. Now, the other challenge we have is feedback inhibition, because if we produce too much methionine or threonine, it stops the enzyme from working, because it says, well, we've got enough now. So we have to get rid of that sort of feedback inhibition. So, yeah, I think we showed this on the amino acid production. So what we have to do is find out which enzyme to mutate. So it's obvious that if we've got feedback inhibition, cut the branch off. Let's cut the homoserine dehydrogenase off, so we're not going to get methionine or threonine. And if we do that, then the carbon goes down this branch, doesn't it? So that's a simple way of doing it, but that's what you have to do. You're kind of channeling the carbon to where you want it to be and producing less of the undesirable products. And because you cut the branch off, it becomes what we call an auxotrophic mutant. So rather than doing a lot of bioassays to see how much lysine we're producing, all we have to do in the case of lysine is get auxotrophic mutants that require methionine and threonine. And we do that by placing onto different agars, one with and one without. And we can say, well, look at this one. This one's where is it? This one, oh, there you go. This one here is an auxotroph. So something happened to its methionine and threonine pathway. There you go, easy as that, isn't it? And then we can also do it when we think about orthotrophic mutants, looking for them that don't grow when the essential nutrient, like methionine threonine, isn't there, and we put it into media with penicillin. Those that can grow, i.e. those that don't need the methionine because it's producing itself, will grow and die because their growing cells are sensitive to penicillin, whereas those that can't grow because they need methionine adding won't grow, and you'll select it like that. Anyway, so that's the old way of doing things. So auxotrophic mutation is one thing, but another way people do it is saying, well, let's just insert the gene of interest. So if we're producing rennet, hemocin for cheese production, look, we don't want to go to calves and things like that. Why don't we just get the gene, ligate it, as we did in metagenomics, and then put it into a new bacteria? So let's see E. coli produce the rennet. It's much easier, isn't it? So this is genetic engineering, and back in the 90s, this was all the rage. People saying, oh, GMO, how can we have this? It's going to destroy the world. I think thankful I did it now, though. And what made possible genetic engineering? Obviously, an understanding of genomics, but also the tools. And one of the tools was restriction enzymes. So restriction enzymes are molecular scissors, and what they do is cut out palindromic sequences. So does everyone know what a palindrome is? Are you going to tell me all this? Well done. It's sequences that read the same backwards and forwards, and that's going to be important when we talk about CRISPR. So palindromic sequences are short pieces of DNA which read the same backwards and forwards, and what these do is act as diets for cutting. So restriction enzymes, and there's lots of them, will cut at different places. So if you know the sequence, you get the restriction enzyme of interest, and then you'll cut it, and you'll cut the gene out and put it into where you want it to be. And there's different ways we can do it. We can do conjugation, which is transfer of plasmids. We can do transformation and transduction. So although these are still relevant, these are still relevant. So in transformation, what we're doing is getting what we call naked DNA, and then we're doing electroporation, which produces holes in our host cell. And when the holes are formed by electroporation, the DNA becomes inserted, hopefully integrated into the chromosome, and away you go. You've got your genetic modification. A lot of people do this. It's not easy, as always, but it's a traditional way of doing things. And obviously in nature, this happens. This is why we acquire multi-drug resistance. This is why we acquire new traits. Now, another way is bacteriophage. So we talked about bacteriophage. Now, obviously the bacteriophage are there to look after themselves, and they'll insert the DNA, and sometimes it ligates in lysogenic, sometimes it will go into a lytic phase. But the copying of the DNA for the phage isn't always perfect. And what they'll do is insert a piece of DNA from the host. So it won't encode for new phage, it'll encode for an enzyme or receptor or something like that. And when it goes to the next host, it'll inject the nucleic acid, it'll become ligated into the genome, and there you go, you've got a new strain, a new property. And that's how evolution, people say, was driven by viruses. This sort of transfer of genes to other cells to build up metabolic pathways, to build up evolution, as I said. Now, conjugation, F plus pilus is an example of that. It's where they exchange the whole plasmid. And people say, oh, your antibiotic resistance, you can put a lot of information on a plasmid, much more than the phage and things like that. So people do conjugation as well. So these are the sort of classic techniques that we call them classic. Back in my day, these were kind of, wow, it's the future and things like that. So typically what you'll find then is that this has been successful in terms of producing insulin, rennets, other sort of proteins, because the E. coli has been a workhorse, basically overproducing these cheap or made high yields on cheap media economically than you would do if you had to do it any other way. So it's been a workhorse, and like I say, progress moves on, and the game changer, what people think is going to be CRISPR. So you've heard a lot about CRISPR. CRISPR's been in the news for the right reasons and the wrong reasons, but some people say it's the ultimate gene editing tool. It literally is, where you can change nucleic acid sequences, you can insert new DNA, you can literally make any sort of change you want. And obviously some people say, oh, this is unethical. Other people say it's a wonder. And anyway, it's what it is. So CRISPR stands for Clustered Regular Interspace Short Palindromic Repeats. Palindromic being the operative word. So palindromic sequences are short pieces of DNA which are read the same forwards and the same backwards. So long story short, what we find when we look at E. coli, where it was discovered, or any cell, matter, what we find is palindromic sequences are between spacers. So these are repeating sequences, regular, interspace, short repeats. So these palindromic sequences are between these, what we call the spacer one. And back in the day, we said this is junk DNA. It doesn't encode for anything. It doesn't encode for an enzyme. It doesn't code for a porine or things like that. And it turned out, and we'll tell the story about CRISPR in a little bit, is that this junk DNA was actually bacteriophage DNA. What was bacteriophage DNA doing in E. coli? And when it comes to think about it, we carry a lot of virus DNA with us, but that's another matter. So what CRISPR actually is a means of cells to defend themselves against bacteriophage attack. And it does this using these spacers. And this encoding for bacteriophages are like our memory cells in our B cells, in that when a phage infects, it recognizes it and it sends out its defense mechanism called the Cas protein. So this gets a bit confusing. So if I go too quick, just say. So what happens is your phage comes along, and bacteria get hit by phages all the time. and injects its nucleic acid material with a view of, we'll say lysogen if you want. Yeah, we'll say lysogens, even though the same applies to lytic as well. So what happens is that the cell knows it's under attack by phage. Now, it's got two outcomes to it. It either knows this phage from before, or it's a new phage. So we'll say what happens if it's a phage it's found before. So what happens is that it recognizes a sequence, and it's got a whole library. 30% of the genome is made up of this junk DNA, this DNA that encodes for phages. This piece of phage DNA, it'll identify, saying, oh, it belongs to this one. Well, green, I suppose I should say that way. So these palindromic sequences read the same forwards as they do backwards. And when they make RNA, this forms a loop because it's reading the same forwards and backwards. Oh. Now, also on that its encoding is this one, which corresponds to the phage DNA, which is infecting it. Now, what it does is get put into what we call this Cas protein. So this Cas protein is a nuclease, scissors, it's a helicase as well. Helicase unzips DNA. So what happens is that the phage comes along, injects its DNA or RNA, and then what happens there? It hybridizes with this Cas protein, and it cuts that DNA. The phage DNA is done. It's basically cut to piece. Well, not cut to piece. It's cut in that particular point. And what it'll do, the cell takes that piece of DNA, puts it into its genome, the prize of war, if you want to call it that. And so by doing so, when the phage attacks next, it's already ready to go. Now, if it's a new phage, what happens is that it injects DNA, but the same kind of thing happens. It sends out its Cas proteins, cut the DNA, and take it and integrates it. So it knows it for next time. These are passed on from generation to generation. This is what drives phage mutation. If your phage didn't mutate, it would be neutralized. And interestingly, in the lab, when you look at phage mutations or resistance to phage, you often see it changes the receptor. But in nature, this is what basically gives normal cells, wild cells, cells in the real environment, protection against phage. It's not to do with changing receptors and things like that. Is that confusing for everyone? And just to finish off then, basically what we have then, we have the palindromic sequence. The palindromic sequence causes this loop, which associates with this Cas protein. And this is the RNA, which is basically hybridized to the target, new phage or whatever. And these are the nuclease activity. The helicase unzips it. And it goes without saying that if you can change, cut the DNA at very specific points by modifying this RNA, which we call the guide RNA, what you have is a gene editing system. And this gene editing system can be used to knock out genes, or it can be used to insert genes, because it's literally having restriction enzymes, but using them in a living cell. And you heard stories about cystic fibrosis. Maybe you've heard the story about sickle cell anemia being cured using embryos. Well, it's not mainstream, but that goes to show the power. And in the last lecture, I'll tell you the story about how CRISPR came about and how the big legal battle is a lesson to everyone who does research. But we'll talk about it at the end. And what I'll do is I'll have a revision class. If you want to come with questions, that's good. I'll bring questions a lot of my own, and we'll kind of revision class then. And we'll finish off this unit.